Okay, I think we're ready to go.
All right. Well, welcome to the 2022 NAFEX virtual conference. Good fruit from the ground up. And so thank you for joining us today. My name is Chris Mannix, and I'm a board member of North American Fruit Explorers, or NAFEX, as I said previously. I'll be facilitating this session. And the session is entitled Managing Pests and Diseases with Minimal Inputs. Before I introduce today's panel, I'd like to share a few housekeeping items and a little bit more about NAFEX as we have people trickle in. First, this is a webinar, so this might be different from some other Zoom meetings that you've been on. All participants' audio and video features are going to be disabled. Second, we encourage you to ask questions. You know, we're a member-based organization, and your questions are really important. And I know that both panelists today will be interested in what you have to say. So please use the Q&A tab as a Zoom feature to ask topical or technical support questions for our co-host. Third, if you're new to Zoom, you can adjust your screen view and some of your Zoom settings on your device. Finally, we're being recorded today, and this will be made available to members right away. And conference participants, all of you, at nafex.org. After about a year, then it'll go up on our NAFEX TV channel on YouTube. So a few words about NAFEX. We were founded in 1967. Kind of as a spin-off of the Northern Nut Growers Association. We're a network of individuals throughout the United States and Canada and beyond that are devoted to the discovery, cultivation, and appreciation of superior varieties of fruits and nuts. Although the ranks of our membership include professional pomologists, nursery owners, commercial orchardists, and more, NAFEX members are all amateurs in the true sense of the word, and we are all motivated by our love of fine fruit. NAFEX members share ideas, information, experiences, fruit propagation material via our website, social media channels, Facebook, YouTube, and other fruit-specific interest group meetings that we have every month, and this annual conference like you're here today. And in the future, we hope to be in person again. So as a paid member, you also get 4 editions of the Pomona Journal each year and an ability to search back 50 years of our Pomonas in our digital library. And that contains a wide array of data, and it's really interesting looking at. The organization that you're all a part of exists because of you. And so we encourage you to continue to support this organization by being a member and also by being involved. Ways you can be involved are by joining an interest group, becoming a committee member. There are a lot of committees to join, and we're trying to build that back as we build our ranks of membership back up. And also, if you're interested, become a board member. It's not as crazy as it seems. And, you know, we are looking for people that are, you know, care about fruit and are willing to get involved. So please visit our website at nafex.org for more information. So anyway, blah, 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 right? So it's my pleasure to introduce our 2 panelists today. And we are joined by Tom Dykstra of Dykstra Laboratories and Tom Murray of Slow Farm. So I'm going to introduce Tom first here, and then we're going to get started. So Tom owns and operates Slow Farm up in Eugene, Oregon. SLOW stands for, in other words, SLO, for seasonal, local, and organic. He has leased farmland for 15 years and undertaken the task of rejuvenating and expanding the orchard in order to enhance production to serve the local market. Slow Farm is located about 6 miles from downtown Eugene and could be considered an urban farm and with intensive production on a small acreage. So, Tom, if you're ready, Tom Murray. So we do have two Toms today, so I'll be referring to Tom Murray and Dr. Tom Dykstra as Dr. Tom. So, Tom Murray, take it away.
All right. Hey, thank you so much, Chris. And I'd like to express my gratitude to everyone at NAFEX for pulling the conference together and all in attendance today for supporting this. It really is a wonderful opportunity, and I'm honored by the invitation to share this time with you and the opportunity to, share my knowledge that I've gained through my experience as a small farmer orchardist over the years. So, and I'd like to add to that that just so everyone is aware, I'm considered a first generation farmer. I grew up in an urban environment, and I've had a lot to learn over the years because I was not born into that knowledge base. Luckily, on my journey, I've had terrific mentors that have helped me on my journey and helped guide me to success. So that being said, yeah, I'm a very much on that urban kind of boundary, really close to my market, hyper local marketplace. I specialize predominantly in palm fruit, and although I do have a handful of berries and grapes, blackberries, and some Asian pears. I guess that kind of falls into the same category as the palm. And I used to grow annual crops, but over the last decade, I have pretty much streamlined the operation just to be focused on the palm fruit production, of which I manage intensively about 4 acres, and it's creeped over just over 1,000 trees now that I'm mentioning, which is mixed production. There was existing orchard, as Chris had mentioned, that went through a number of years of regenerating. Based on when I came to the situation, it was very neglected. So we've had to rejuvenate that orchard. Those are kind of traditional freestanding open center trees, and then I have expanded the orchard into more of a modern intensive central leader on M26 rootstock. So I have both kind of models, the old and the new, in my production system. However, being a small farm, we're still really focused on doing a lot of manual work, intimate work with the trees, with our hands, rather than everything being mechanical, as so much has gone that direction in the last 10 to 20 years. But the purpose of our discussion today was going to be specifically on pest and disease management with minimal inputs. And I can share what I've gained over the years. Like I was just saying, that a lot of the work that we do is really hands-on and intensive. And so I think that really pest and disease management, as I've come to know it, really starts with good cultural practices. And all of the literature will always come back around to that. That first and foremost, what you could do is make sure that your soil is healthy, and that your trees are in good health, through pruning and, all those cultural practices, pruning, thinning, and sanitation. So that being said, the intensive aspect of the orchard is really, a lot of people approach it as far as getting really close to their trees and working with the trees. It seems to be more of an annual basis when they do the pruning in the wintertime, pretty much in dormancy. We've been practicing pruning twice a year. So we're pretty much coming to every single tree and working with them in assessing the needs of that tree and trying to cultivate the tree in such a way that it's going to encourage vigor and healthy fruit buds, fruiting wood. And so we're doing that every 6 months. So summer pruning is just as intensive and serious for us in our management strategy as winter pruning, which a lot of people don't really practice, and they're a little unfamiliar with summer pruning techniques. And it took me a number of years to kind of learn that from my mentors, but it's really critical to this pest and disease management, because it's really comes down to what I identify as, the three aspects of what the tree needs, and penetration. The things that you need to be most concerned about is adequate airflow, light penetration, and then of course spray penetration if you are using any sort of biostimulants or crop protectants. And if everything is dialed in, first the foundational piece being the soil health, then you should have pretty vigorous growth, and that's something that you really have to manage, because very quickly trees will become so bushy that those three factors start to get restricted. You're not getting much light in, you get a lot of shade inside the tree and on the limbs, which actually intense shading with no sun penetration starts to make the bark kind of a little bit weaker. The cell wall becomes kind of thin, and that's where you often find disease. It's easier to penetrate, and you also find that it makes really great nooks and crannies in there in the shade where a lot of pests like to hang out. There's a number of pests that are really deterred just simply by the sunlight getting in. They want to be hidden and in the shade. So that's a really critical factor, is this the pruning, and we'll share some slides here in a few minutes to kind of show you what it looks like in both seasons, in the summer and the winter, so that you get kind of an idea of how important it is and how much we take off. And then, at that time, you're really close into the trees, and you got your eyes really tuned in to all of the branches and everything. So you can really see where there's problems, where there's, say bark necrosis that needs to get cut out and disease is starting to really proliferate. Or you can find, where there may be insects that are kind of entrenched in the canopy, that you could simply go in there, take some buckets, or disease. We've been experiencing these ongoing warming and drought cycles here in the West. In my particular zone, which is maritime climate, one of the diseases that we haven't dealt with a whole lot in the past, that is starting to creep in and become problematic every season now, is powdery mildew. And that's another disease that you can really get a good handle on it as long as you're out in the field and policing for it and pruning it out and removing it from the field, in buckets or something like that, not dropping it on the ground. So again, the sanitation becomes critical in managing those types of those fungal diseases. That being said, as I was mentioning before, it really comes into, the sun and the airflow, and then also spray penetration. And so I do use some biostimulants. I've been using biologicals like compost tea that we make on the farm for years. And I use that both as a soil drench and as a foliar application, as well as for managing particular pests. I use some biologicals. And again, if the spray can't really penetrate into the tree, especially with biologicals that need to get right where the pests are so that they're come in contact and ingested, they just don't have a high residual in the field or efficacy time. Biologicals, they're living agents out there, bacteria and such. And so they they degrade quickly, especially in the peak of the season when you're getting a lot of degree days that the sun's really high, it's hot, it's drying out, so if you're going to be utilizing those kind of tools in your management strategy, then it's critical that all of the parameters are set up for those agents to be used with the highest success. So yeah, and that being said, I think maybe it'd be a good time now to let's go to the slideshow so we can look at some of the pruning that I have.
I would just like to say, Dr. Sandy asked, Tom Murray is in Oregon, and he's mostly speaking about management of palm fruit.
Back to the beginning here. Yeah, okay. So we're going to start with, there I am out in the orchard in the fall. And yeah, the next slide. So this is going to be some of the trees. This is in the older block, the orchard that this is probably about the 10-year mark into the regeneration of the orchard, of which more than half of the original orchard was entirely removed because the neglect was so bad that the disease was so prolific. And some of those trees we tried to bring back with intensive pruning to try and stimulate them, and they just never really came back. They just kind of floundered. Some of them were dying out. So we removed a lot of those and replanted a number, but this was one of the original trees that was there that came back quite nicely. So you can see this is just after summer solstice, which is kind of the time when I like to begin summer pruning. That's kind of coming up on the backside of thinning. So you have that first full spring extension has kind of reached its peak. And at this point, I like to open them up because that's when they've really bushed out and become very thick. As you can see, the shading under the ground, there is some sun right there, but it's becoming very dark under the tree. There's a lot of shade going on in the canopy. So if we go to the next slide, that's going to show that's after pruning that particular tree. Looking back at the slide, I'm like, wow, we left it kind of bushy. And sometimes these photos don't really do justice to what it looks like on the ground, or if you were kind of up in the tree. But really, we kind of opened it up so that sun is bouncing around throughout the tree. I like to be able to see into the center of the tree. That's kind of what when I'm teaching people or teaching the people who work for me, is that if I'm just walking through the orchard and I can't see the main trunk really up in the canopy and those lateral branches extending out from the center, then we haven't pruned enough. I really like to be able to see into the tree, and then I know that when I'm coming back through and using some of the biologicals for management, that they're effective. And then also light is bouncing around inside the tree, which is going to increase the color as well. So then looking at this tree before and after that summer. So if we go to the next slide, this is about 6 months later. This is winter, just prior to pruning, to show you what the growth extension was like, the final growth at the end of the season. And so if we go to the next slide, this is what pruning looks like after winter pruning. So you can see we took off quite a bit, and maybe we could toggle back again just to get a good idea of how much comes off. I think the winter photos are a lot easier to see between the summer photos. Yeah, so it's a good amount of growth. And one of the things that I'd like to highlight here is that pruning can be, it's very stimulating the tree. It can be very stressful at times, depending on how you approach pruning. One of the advantages of pruning twice a year is that you kind of regulate the stress on the tree and the production of hormones, excessive hormones, when they are responding to intensive stress. So sometimes what I've seen a lot is when people only prune once a year, they tend to come in and be pretty extreme. And then when the trees wake up in the spring, they have an excessive amount of vigor and they put so much energy into regrowing what was taken off, and it can kind of throw things out of balance. So it's one of the key benefits of both summer and winter pruning for us is maintaining an adequate amount of healthy fruit buds out there, not removing excessively so that you would fall into some sort of biennial bearing, but to keep it really balanced and low stress on the tree, so you're not getting these excessive growth where the tree is trying to make up for what was lost at the expense of fruit production, that causes you to kind of slip more into a biennial bearing habit. So it kind of helps regulate the energy of the tree overall. And at the same time, summer pruning reduces the load of winter wood that's coming out. So that's less that you have to deal with and removing from the orchard. And hopefully, if you're not letting things get too big, a lot of what you're dropping off in the winter is easy to go through the mower or through the chipper, because it's not a huge amount of wood that's a really thick diameter. So let's go to the next slide. Here's another tree. Again, these trees are about 35 years old. And so that's before summer pruning. And then the next slide is going to show you, this is after summer pruning. So you can see with that one in particular, if you go back and forth, there's a good amount that was taken off of that tree. And that tends to be, you know, every tree is treated a little bit differently. They're their own characters, especially in this block with these older trees that we kind of rejuvenated. I didn't train them from the very beginning. And you get to know them over the years. And, you know, sometimes you take a little bit more off in the winter, sometimes a little bit less. But I always know that it doesn't have to be perfect, because, you know, when I'm teaching people again out in the field, I say, well, we'll be back in about 6 months. And, or if we're thinning, is also an opportunity to prune. You know, I basically tell everybody that every time we come to the tree to do anything, we should always have our clippers on us or our loppers nearby, and it's another opportunity to prune. You know, so when we're thinning, you also can prune a little bit to open up some light and get in where you need to go. And then you'll start to see, once the trees have leafed out, where you kind of could have done a little bit better of a job. Maybe it was the rain came early that day and you kept moving for whatever reason, you know, you just didn't really get it fully, you know, to where it needed to be in the winter pruning. But it's okay, because you can make up for that. It's not going to be another year before you revisit the tree. Next slide. Yeah, there we go. So this tree, I threw this photo in there because this is a really good example of what the thinning should look like when you're done. As you can see, everything there is spaced, you know, roughly about 6 to 8 inches apart. This is in our intensive block. One of my dear friends and mentors, who actually was a palmologist and a surfer in California in the 60s, he always liked to teach people with just that it's got to hang loose. This is the measure that he used out in the field, and he would give this to everybody without giving them any technical terms or anything. As far apart as you can spread, you know, your pinky and your thumb. He's like, that's how far you want all the fruits to be from each other. And that has worked really well at helping people out in the field when they're asking me, is this right? Is this right? And then, you know, you kind of give them that, and then they can start to check it themselves. And so if we go to the next slide, this is going to kind of show, this is the same block in summer. As you can see, very well thinned. These trees are in year 5. So, and that's like looking like about 2 to 3 weeks away from harvest time there. We get a lot of cool, cloudy mornings that could linger on here. That's our maritime climate in Western Oregon. Yes, this is a summer day, and it's probably about 55 to 60 degrees out there at like 9 in the morning, but the palm fruit really thrives in this climate. It doesn't get too hot. We get just enough degree days to really get good ripening by September, October. Some late varieties push into November, not too many. So let's see here. I think the next slide is going to be new things. Well, there's the finish actually. Yeah. So that's kind of seeing from thinning to ripening. That's Liberty in the box, ready for market just after pick. And you can see they're a pretty good size and really excellent color, good pruning and thinning, and all those factors alone, you should be able to achieve really good color. If you have an excessive growth, which could be due to excessive nitrogen as well. I experienced that in an orchard that I helped renovate, where we had used the prior farmer was doing a very old school and just using chicken manure at a particular rate every single year, and that's how he had learned it. And the color was terrible, and the ripeness wasn't really didn't really want to ripen, and storability wasn't so good. And then I convinced him. He was very reluctant to say, hey, we need to kind of step off of this heavy nitrogen. I think there's plenty, and maybe we could go more of a biological approach and really just focus on the minerals and the biology and not be worried too much about nitrogen. We turned that orchard around and got some of the best packouts that have ever happened out of there, and the color just went right through the roof. It was just beautiful, the crops that came out the subsequent years. So that's kind of an example of the quality that we're kind of pulling out of our orchard operation now. If we go to the next slide, here's some of the Honey Crisp. I get proud of those, as so many of us do, especially when it doesn't have bitter pit, which has been pretty good. And I also like to include this photo so that you can see, again, you'll notice the understory. I don't keep the rows completely weed free. I'm not too concerned about that, actually. I come from more of a biological approach to it all, kind of an agroecology, which says that, the ground cover, is there for a reason, and the soil needs to be covered, and it's good for the ecology. And, it is astonishing when other more conventionally minded people do come out to the orchard and they're shocked by the understory. And then they're also even more shocked when they see the quality in the box. They're just like, it doesn't make any sense. And it's, I don't know what to tell them. Maybe we'll save that discussion for another time. Let's keep going, please. So here's some of the tools that we use. I included sprayers. So this is a sprayer rig that we built on the farm specifically for handling the biologicals and the compost tea. This is checked basically with the small 5 horsepower impeller motor there. You can consider it microbe friendly. It doesn't get to extreme PSI, and the centrifugal pump on it doesn't cause the microbes to get destroyed as they're going through it. So it's a 200-gallon tank. I can do 1 acre at a time soil drench application, or 2 acres at a time foliar application with the biologicals. But the biologicals, in order to achieve the benefit of the biologicals, only go through this sprayer as far as the fertility and the biology is concerned. And then if we go to the next slide, this is my little mini air blast sprayer. Now I do use a biological through this specifically for the coddling moth management. I use granulosis virus for that, and it works through this. Again, you can dial the pressure appropriately. to work through this machine. And this is the smallest air blast sprayer that you'll ever find, that I was able to find, that actually will go to those low pressures. But I don't really use this very much for the compost tea applications. Anything else that I need to do goes through this, and it's really effective. And of course, I have to show you my lovely 1994 Toyota pickup truck, which lives on the farm and does so much work. It is incredible, and couldn't do it without it. So let's go to the next slide here. Some things that I'm excited about, new happenings on the farm. These are peaches in the greenhouse. This is a new crop that I'm looking at doing. Again, the greenhouse is really to prevent disease. It's for disease management more than, say, early ripening or any of those benefits that you might read about traditionally in the use of greenhouse. And in our climate, in Western Oregon, the maritime climate, some of these diseases can be really nasty, and that's why peaches aren't really grown here, or we're not known for peaches in Western Oregon necessarily. However, having them in the greenhouse has been just night and day. It's actually made this a very viable crop for my operation, and with very minimal inputs outside of the greenhouse. So again, strictly biologicals is all I'm using on this. In the greenhouse, I don't have to use any other crop protectants to try to manage or mitigate disease problems that a lot of the growers who are growing them outside are using very intensive spray programs that I could never really justify in our operation. It's just not by my principles that I farm by. So this has been a really successful project, and the customers are really excited about it. And I wish that I had done this years prior. If we go to the next slide, very proud of this. My first real peach crop came out of the greenhouses this year, and the trees came through great this season. Fruit quality was excellent, again, with very minimal inputs. And that's what I'm so excited about this. I would have never done peaches before outside just because the inputs required, particularly in our climate zone, are so challenging. So I think that kind of takes me to the end of my presentation here. Which is, this is, here we are at market, our local market, as Chris was saying. This is 6 miles from us. The market has been operating at this location for about 40 years, almost. Lane County Farmers Market. I've been a member there for 20 years. I currently serve on the board. It's a real joy to participate in such a community space and to provide for this community for this long. And yeah, this was 2019, which was a really good season. And that's my good friend Dana McWayne, who he was a high school kid when he started working for me. He worked for 4 or 5 years, and now he's an organic inspector. And so it was a joy to work with him and many other people who have helped me in this along the way. And there I am out in the fall orchard there. My pink lady trees. I was really happy with that crop. They came out really, really good that year. So, yeah. There's any
awesome
questions? Yeah.
Thank you, Tom. Thank you.
Certainly.
I see somebody's throwing heart emojis and clapping emojis and festival emojis. There was a couple questions I just wanted to shoot to you from a couple members here, and then we're going to get on to Dr. Tom. So any other questions after this, then we'll just answer it then. So feel free to keep adding your questions in. But Andrew asked, he's in zone 5, and he asked, he's trying to figure out the best times to do summer and winter pruning. And could you address that real quick?
I could, yeah, that's a good question. You know, there's a few different ways to think about it. You can really think about it in terms of the sun cycle. Or, you know, like I said, around solstice or just after solstice and that bud extension. You know, in the literature, with most varieties, there's about 4 growth extensions if you watch your apple trees very closely. from bud break through, basically the last extension happens for us in about the first week of September. You might see another little spurt there, and that could go on for 3 or 4 weeks. Again, that depends on the vigor of the tree, how much care you're giving it, and what the season is like. If it's a really stressful season to come through, we've had some stressful seasons with the drought where that growth has been checked. But in really good growing seasons, you get about 4 growth extensions. Now, the reason I mention that is that I like to prune somewhere just after the second extension, which puts you right around summer solstice or just after that. You have a spring extension, goes on for about a month from bloom, post bloom. Then they're going to break for a minute. That's kind of when we're thinning. And then all of a sudden you have another spurt that happens. That's a pretty good extension going into summer. And that's kind of like, I like to prune kind of as that one's wrapping up. And another way to think about it too, is that if you prune too early, go and summer prune too early, you'll get a good amount of regrowth. Now, when trees are young, actually that might be desirable. It's a really good time to be shaping the trees in the summer. You want some splitting and more bud sets or whatever. But with older trees, if you're trying to open them up for sun to get in and for spray penetration, I learned this the hard way by kind of chomping at the bit and trying to get ahead of my season and all my tasks and going in and pruning early. And my mentor was like, if you prune too early, you're going to get secondary growth. And I was like, oh well, whatever. And he was right. Give it about 5 or 6 weeks later, you look back at the orchard, it looked like we didn't really do much. And if I had just waited about 2 to 3 weeks, 2.5 weeks, maybe it would have been a different situation. So it's really the summer pruning. Yeah. Somewhere around, I kind of like to have it wrapped up basically, and it rolls into the general task of when harvest is coming in and all those things. I look at it kind of in a calendar schedule of tasks, and I try to have it all wrapped up by like August 1st. So I'm going in about June 15th, starting early. That puts us right around solstice. We're really getting into it. And then we've got about 5 weeks right there to just get it done. And then we're on to the next tasks. Winter pruning, as far as it's like anytime after dormancy sets in, and try not to push it too late. The earlier you can prune, I feel like the better. Depends on where you're at. I know that kind of in the Northeast, where they get some really serious cold weather, they end up having to wait because some of those varieties and rootstocks are going to get winter damage. So they push it up towards the end of the winter season, and that puts a lot of pressure on to get it done really quick. So I don't know if that's helpful at all, but basically wait for dormancy and then after the second growth spurt.
So a little one last one here, and then we're going to switch over to Dr. Tom.
Yeah.
So Mark comes from the other side, Mark Walbers, and he I'll preface this. Well, I'll say it first, but does summer pruning in a maritime climate open the trees up to fungal infection? And I think you and I both have strong opinions on not winter pruning prunus, because it will kill a peach or a plum or a cherry very, very quickly that way out here, especially. But could you speak to that just real quick, just maybe just a few minutes, and then we got to shift over.
Yeah, I think that the best, the way to approach that for me, because you do have to prune, and summertime may be your best time, specifically with prunus. Again, be ready and follow up. My strategy would be ready, follow up with the biologicals, the compost tea, to bring the balanced, microbial diversity, right to the site where you're pruning, because that's going to be your best bet. And oftentimes that may be what's missing, is that you got to get the microbial quorum there. So you've got to get a good diversity of microbes for the tree to be able to even initiate its own immune response. Yeah.
Cool. Thank you, Tom. We definitely have some more, but I'm going to shift over to Dr. Tom here.
Yeah.
And we'll be coming back to you in a little bit.
Excellent.
Okay. So our next speaker, Dr. Tom, he is the owner and laboratory director of Dykstra Laboratories in Gainesville, Florida. And he serves as a scientific advisor for advancing eco-agriculture. For 25 years, he has studied electromagnetics, how electromagnetic fields affect life, especially as it relates to insects. He holds entomology degrees from Cornell University, as well as the University of Florida, and has awarded 7 patents with 1 patent pending. Dr. Dykstra consults for farmers, agricultural companies, tech firms, attorneys, international governments, and insurance agencies. He has visited 3 continents and presents lectures on diverse topics covering entomology, olfactory physiology, biophysics, paramagnetism, neurobiology, and biological antennae. In terms of active research, Dr. Dykstra deciphered the insect olfactory code back in 2016 and characterizes chemoreceptors for various medical and agricultural insect pests. He teaches both farmers and agricultural consultants how to raise healthy crops for their families and for a profit. So with that said, go ahead, Dr. Tom, take it away.
Well, thank you. Thank you. You are one of the few people who have actually pronounced bioelectromagnetics correctly on an introduction. So congratulations to you, Chris. Tom, great presentation. I was ready to hand over the rest of my presentation to you because I enjoyed not only listening to you, but you just seemed to be hitting all the buttons and saying all the right things. So thank you so much for your presentation. I'm now going to attempt to, let's see, I'm not there yet. Hold on. I'm going to attempt to share screen and see how this goes. Hold on. Now we're good, right?
All right.
Okay. Yes, it's just a matter of outsmarting the computer. I mean, that's all this is really about. So, all righty. Title of my talk, fruit pests and their food preferences. So I'm going to be covering, of course, insects. I mean, this is mostly my background. Bioelectromagnetics too. Won't be talking too much about that, but I will be talking about the insect side. Of course, we're all dealing with fruit pests. We have mostly oranges down here in Florida, but there are many other types of fruits. This is all applicable to any type of fruit tree, really any type of plant. And what I'm going to do is I'm going to discuss that, hopefully open up your mind for those of you who haven't heard anything like this before, and we'll see if we can make some progress. So, all righty. Here's the shocking comment, which I like to start with because I figure if I'm going to lose someone, you might as well lose someone on the first slide. Insects only feed upon food that is considered unfit, nutritionally poor, dead, or dying. There are no exceptions to this. And so because of this, it changes your whole mentality about how you are looking at insects because they do have a simple digestive system. There are reasons why they do what they do. And because not all insects are the same, some of them will be feeding on dying plants, other ones will be feeding on dead plants, others will just go after nutritionally poor ones or unfit ones. All the insects will have their own degree with which they work. So if you have grasshoppers, for example, and you're given a choice between unhealthy food or healthy food, the conventional wisdom will say that the grasshoppers will go equally from the unhealthy food to the healthy food. And this is actually not what happens. In fact, the grasshoppers or any insects, they will always go toward unhealthy food. This is their modus operandi. This is the way they wish to feed. And this is something which many people have observed over decades. And I'm just going to be really just putting some numbers to it. So we take a look at fruit flies. I'm sure fruit flies are probably in a lot of the fruit orchards that are out there, like Drosophila suzuki'i has certainly taken prominence, but fruit flies are found around overripe fruits, vegetables, and other organic foods that are damaged, fermenting, or in an otherwise state of decay. So even Drosophila is going after this. We don't find them going after healthy food. There are some cases where people will, it will look like they're actually going after healthy food, but a quick test will tell you that it's not. So here is food right now, which is more likely to attract Drosophila than food that actually looks good. So we see a lot of damaged fruit. There's going to be an odor coming from them, and these will attract Drosophila. We should be able to know this. This is common sense already, because if we're talking about plants, and that's what I'm going to be focusing on today, if we're talking about plants, you know, why haven't the insects destroyed planet Earth? They should have. The reason why they should have is because there are no barriers, there's no nets. They can go around. Their rate of fecundity is tremendous. They can destroy a tree, defoliate it very quickly, and they should have overrun planet Earth already, but they haven't. And here's some good evidence right now. When I was in Kauai, you can see the massive amount of vegetation that is here. I saw lots of plant-eating insects while I was there, and yet most of the plant life, most of the vegetation was left untouched. So this does have to get you thinking as to what is going on. So in plants, we have the very well-known phenomenon of photosynthesis. We know that the energy from light is absorbed by the chlorophyll, and there are various types of chlorophyll with various frequencies that each one of them will tune into, but all the chlorophyll are absorbing, for the most part, visible frequencies. And with the addition of carbon dioxide and water, 6 parts each of those, the plant, using the energy, will be able to produce a molecule of glucose, which is sugar. It'll also produce 6 oxygen molecules as well. But this is how all plants work. And so all of this is happening through the leaves, almost predominantly. You don't see too much of it occurring in the fruits, the stems, and certainly not the roots, but you do get a lot of energy production or sugar production in the leaf. So when you're looking at the orange, which is set before you right now, this is not an orange which is photosynthesizing right now. All of the sugar that is inside it came from a leaf. And so because the leaves are the important absorbers of this energy, it's good to focus on these leaves to understand a little bit better how a healthy plant works, because a healthy plant is going to be photosynthesizing well, and an unhealthy plant will not be, which means we now have a way of measuring it because we understand one of the byproducts of photosynthesis is sugar. So we have monosaccharides, which is a 6-carbon sugar, disaccharides, which is 2 6 carbons together, trisaccharides as well. The monosaccharides, we mostly have the first and the third, fructose and glucose, around the plant. In the disaccharides, sucrose is the main sugar that you will find mostly traveling through the plant, especially the phloem tissue. And this is just composed of a fructose and a glucose molecule. So these are the types of sugars that are in the plant. They come from photosynthesis. They cannot be pulled up through the roots to any degree. And so the vast majority is going to be coming in through the leaves. And so we've now got a way to take a look at the health of the plant in order to draw some conclusions about plant health. So the quickest way, there are many different ways to do it, of course, but the quickest way is to determine plant health is to test the plant sap with a refractometer, and that would involve crushing the leaf, sometimes 2 leaves, 3 leaves, or more leaves, and getting the sap. It's not just the sap as the phloem sap that I'm referring to. I'm referring to all of the liquid. So that would include anything in the phloem tissue, the xylem tissue, the apoplast, epidermal cells, mesophyll cells. All of that liquid is crushed, and we get several drops, and we put it in a refractometer in order to understand how healthy the plant is. The brix refractometers, there are 2 main types that are out there. One does not require batteries. That's the one off to the left, and there's a little view plastic screen which you can lift up and place a couple drops on it and look through it, and you'll be able to get the reading. We also have digital refractometers. These are the ones that I prefer to use because they're faster, easier to clean. And the digital one off to the right does require batteries, and even has a temperature compensation, which is sometimes important. It doesn't make a huge difference though, but it does make some when you're dealing with the Florida climate. So these refractometers will give you the information that you need, but we do need to get that plant sap, or that liquid portion of the leaf, because the leaf is where all the action is occurring. And so you can use a garlic press. Many people do. If you're a homeowner, the garlic press is often the one of choice. They're small, They don't handle too many leaves. You can have 1 or 2 leaves in there and it can get filled, but it doesn't handle as many leaves as I would like, but it can work. And the garlic press I still use for testing fruit, not the leaf bricks, but I still test fruit relatively routinely from a garlic press. That's just fine. But if I am asked to go to an orchard, and I got 100 samples to do, I'm not going in there with a hydraulic plant press. So we got 2 different types of hydraulic plant presses over here. And depending upon the size of the leaf, you can easily put 5, 10, 15, sometimes even 30 leaves, depending upon the size. The one off to the left can handle more leaves. The one off to the right has a smaller compartment. But each one will do the job in order to squeeze the leaves and get us the plant sap that we need in order to test it on a refractometer. So moving quickly between them, if I'm asked to scan a field, is really the only way to do it is a hydraulic plant press. However, I do want to stress, though, that for a lot of the homeowners who are listening to this, a garlic press will work out well. You do need to be strong hands, and you will get tired over time if you're doing 100. This is why I go to the plant press. But if you don't break the garlic press will break over time. You want to go ahead and go that route. That'll be the easiest and the cheapest route to go. So we're taking a look at this in part because the plant is producing sugar. It's almost all of it's coming through the leaves. There is some photosynthesis, of course, in the stems, the petiole a little bit, but not much. But about 50% of the plant sap sugars can vary between 20 to 70%, but about half of it, maybe 40 to 50%, is going down to the roots, and that's to feed all the microbes. So all of those microbes that Tom Murray was talking about before and putting down biologicals need to be fed, and the plant will take care of that, and that's part of the reason. So you do want to know how healthy the plant is, because it's not only got, let's say, fruit in order to make, but it's also got to be feeding the microbes, which are going to further redound to the health of the plant. So with that, there's a chart that I have made up, and this has to do with leaf bricks. This is not root bricks. It's not stem bricks. It's not even fruit bricks. It's leaf bricks. So I'm just looking at the leaves. I want to understand how healthy a plant is. And when we do that, I've got 4 main categories that I've been able to delineate. And the first one is essentially leaf bricks of 1 to 2. They're not healthy, as you might imagine. There's not a lot of sugar, not a lot of photosynthesis. Force feeding is necessary for these type of plants. Many house plants can be 1 to 2. A lot of the greens on golf courses will usually be 1 to 2 as well. And if you have any plants in nature that are at 1 to 2, they'll get picked off by insects and disease very quickly. They need to be fed pretty much every day. This is why, like on a golf course, you see them feeding the stuff every day in order to keep them alive, because if you let it go for a week, it's usually dead. But these plants don't do well on their own. Obviously, they're at 1 to 2 bricks. But the next part, they're doing much better. These guys all have a fighting chance. This is about 3 to 7 bricks. And this is where most of the crops in the country are grown at. They got a fighting chance because they look okay. They grow, they do pretty well. They're not doing too bad. And this is the second level, and they don't need to be force fed. Maybe they just need to be fed once a week. But an important thing happens during this phase, and that is at about 6 bricks. The plant's secondary metabolites increase. So as you go from 6 to 7 to 8 to 9 and so on and so forth, you get a massive increase of plant secondary metabolites. These metabolites are adding color to the plant, smell to the plant, taste to the plant, which means if you have anything below 6 bricks, it's going to taste like cardboard. And so you're looking for something that's going to be at least 6 bricks, because that's going to give you the flavor and the look that you wanted, like some of the fruit that we saw earlier with Tom's presentation. So this only begins at 6. You have virtually no smell. I've tested tomatoes, for example, at 3.9, and I mean, you cut it open and you couldn't tell that it was a tomato. There's absolutely no odor to it. The next stage, the third stage, is roughly between 8 to 12. There's a sword and shield. And that's because these plants are really pretty good at defending themselves. The plant's secondary metabolites have started to increase quite a bit as we're going from 8 to 9 to 10 to 11. And because of that, they're able to defend themselves against insects. They're able to do a pretty good job of defending themselves against all aggressors, even disease as well. And so the water retention abilities really increase at this point. So if you're in drought conditions and you've got plants that are between 8 to 12, you're going to be losing a lot less water, maybe 25% less, in some cases 50% less water if you've got a high brix plant, because that usually means there's a lot of microbes in the soil that is also contributing to the health of the plant, and they're going to be able to hold on to that water so much better should you find yourself in drought conditions. It's not much of an issue here in Florida. We almost have too much water, but of course, other parts of the country, California being prominent right now, and of course, New Mexico and Arizona as well, do need to hold on to that water. It is in this stage that insect resistance really begins in earnest. So brought from 8 to 12, a lot of insects just have difficulty with these plants. The last one, these are all objectively healthy. So if I'm getting a leaf brix of 12, 13, I prefer to get to 14 because there's always going to be fluctuation in the leaves, so that if it goes down to 13 or 12, you're still relatively well protected. But they're objectively healthy. They're objectively healthy. There's really no way that would describe it. These plants are doing so well that there are no insects attacking it. There is no disease, are no diseases that are attacking it. Food, in my opinion, is now being produced. It is now fit for human consumption. It's the type of food that we want to be eating for a good, healthy, long life and to avoid diseases ourselves. And this is something which we are all looking to attain to. So on this chart, I've now superimposed various insect groups so that you can see where they stand. And as you can see, most of the insects, they're pretty much done by about 12 brix, with the possible exception of the grasshoppers, who continue to impress me more and more. But the grasshopper group will leave. I'm suggesting brix levels at which insects lose interest in plants. So at about 10, 11, or 12 brix, the grasshoppers will start to lose interest in a plant, and they will stop biting, and they will stop eating the leaves or doing whatever they're doing. And so this is what has been noticed. The grasshopper groups include grasshoppers, the katydids or longhorn grasshoppers. It can also include the grillids, crickets as well. And beyond this, though, this chewing group, and I had to separate them, the next group of chewing insects includes pretty much all the rest of the insects. And these include the Lepidoptera, the butterflies and the moths, Coleoptera, the beetles, the Diptera, as well, the flies. These groups that are often more or less chewing insects, although some of the Diptera do suck. There are some, maybe literally and figuratively, but I'm talking about insects that are actually chewing on the leaf, chewing on the root, chewing on the flower, chewing on a fruit. They're going to leave it a little bit earlier. Once the plant gets to about 9 to 11 brix, they will lose interest in the plant as well. And so then we move on to the next level, and we've now got two groups of sucking insects, the aphid group and then the rest of the sucking insects. I have had to separate them because the grasshoppers in the aphid group have separated themselves, but the sucking insects, they will actually start to lose interest in the plant at about 7, 8, and 9 brix. By the time you get to 9 brix, you have virtually no sucking insects on the plant anymore. But you usually find them below that. The aphid group is a little bit lower, about 6 to 8. Often when I'm out and I see aphids attacking a plant in earnest, certainly hundreds of them, maybe even 1,000 on a plant, I know I'm going to be looking at a 3 to 5 brix plant. And that's just the way it always turns out. So the aphids are there really going after the lowest quality plants. So these groups are just the beginning. There are 1 million species of insects, but we get an idea. I wanted to keep it as general as possible because too much detail can get to be a bit confusing. But on the other hand, I am talking with a bunch of fruit growers, so I'm going to have to talk about a little bit more details now, and so I will. And so what I'm going to do is I'm going to talk a little bit about citrus. I'm talking about citrus in Florida. Even though this is applicable to any other plant, doesn't matter whether it's peaches, whether it's apples, you've got a group of insects that follow the group that those 4 major groups that I just mentioned. So on citrus trees, if you've got a relatively healthy plant and it starts to dip below into that unhealthy level, which is easily measurable with the refractometer, it will start to attract grasshoppers, katydids, and even crickets. So mostly the first two. So here's a grasshopper picture eating a leaf, doing what it does best. Over to the right, I have a citrus plant. The citrus plant, as you can see, especially with the middle leaf right there, has been eaten by both grasshoppers and katydids, which have been found on this particular plant. Some of the other leaves are looking pretty good, but the more weakened ones are, of course, the ones that are being attacked. And you can see that this is kind of going to be the first stage attacking the citrus. So as we move on down the line, I'm now going to talk about other citrus pests. Remember, this is applicable for anything, apples included, but I'm just going to be talking about these insects. We have the giant swallowtail, which beautiful butterfly on the left, a papilionid, but on the right, the larva looks like bird poop. And it is also known as the orange dog caterpillar, in part named because of the osmeterium, which is kind of a reddish or reddish orange. And Papilio crescentis is a relatively small pest on most citrus. You will find them attacking, and they're going to be found on slightly lower healthy plants than you will find the grasshoppers, but they will appear. And then once the plant usually gets to a lower level, they will start to disappear as well. One of the next ones to come in is Diaprepes abbreviatus, which is the citrus root weevil. First made its appearance in Florida, 1964, soon after high density planting was begun. It was such a pleasure listening to Tom Murray's talk when he was talking about making sure that light got through, because if light does not get through, and if you have high density planting, for example, which is unfortunately more common than it should be, the trees will suffer. The roots will intermingle, and once the roots start to intermingle, they get weakened, and then Diaprepes abbreviatus moves in, and it becomes a problem. So it was introduced in '64. This is one of the next steps that happened in citrus. After that, it was 1993 when the citrus leaf miner came into the picture. It took another 7 years before it came to California in 2000. But obviously, this is just a classic leaf miner attacking a leaf. It can attack. This is not just an orange citrus tree, but of course, it can attack many other types of citrus as well. And this would then be moving on down. These are still chewing insects, though. They're still chewing insects. But now we've moved on to a sucking insect, and the Asian citrusillid is a true sucking insect. Once the plants, or the trees in this particular case, move down health level, they'll start to attract more of the sucking insects, and then disease is more likely to be propagated. So the Asian citrusillid was attracted to the orange trees in 1998. Of course, it was present long before that, and it does spread citrus greening, which is a disease which is able to be spread in only the weakened trees. The stronger trees are able to fight it off pretty well, because we still do not have 100% citrus greening, and if we do, some of the trees act like it's not even affecting them. So some of the trees are absolutely doing well, others do not do so well, and we have lost a lot of citrus trees, in no small part, as you've probably heard, due to the Asian citrusillus. So this is kind of where we are right now. We're kind of in this stage of the downfall of the citrus in Florida, and we've obviously lost quite a bit. The newest one has come in. The mealybug, which is in that aphid group, has just been introduced relatively recently, and it's starting to make its way around. It's about 2009, when it's either called the Lebec mealybug or the spherical mealybug, but Nipea is its name, and Viridis, and it is now attacking citrus as well, which means the citrus is reaching the point where it's so unhealthy that the insects that are now coming in are going to be able to feed on it, because they've got the interest and the ability to feed on what really is amounting to a low brix plant. Easily measurable with the refractometer. I can tell you that virtually all of the trees, with few exceptions, that we have measured in Florida citrus are 7 and below. We almost never reach 8 or 9. I have reached 9, I have reached 15. There are exceptions that are out there, but for the most part, virtually all the citrus is 7 and below, and is therefore very susceptible to this. Once you reach 9, a lot of these insects disappear, and with the 15 brix that I just mentioned right now, there was no disease and there were no insects that were attacking it at all. It was completely clean. So we understand that this is the process and this is applicable to all plants. I just happen to use citrus as an example because this is a fruit growers kind of get together. So I wanted to focus in on that, although I understand maybe a lot of you are not from Florida. If you are, this is more applicable. If not, there are going to be a different group of insects. But even though there's going to be a different group of insects that may be attacking, let's say apples, which are on the opposite side of the country from Florida, you're going to have the same groups attacking the apple, just like they would as they are doing in Florida. They're going to be different insects, but they're going to be following the same groups and the progression will be the same. So, all right. So just to review real quick, I just wanted you to see, these are some of the insects that I talked about on citrus. Napiacoccus has just arrived on the scene. That is part from the aphid group. Diaphorina, which is the Asian citrus psyllid, was a sucking insect. That was in another level of insects. Papilio, Diaprepes, and Phylognistis are chewing insects, and they will be found on a slightly higher brix plant, and we find them on plants that are doing a little bit better. Obviously, Napiacoccus and Diaphorina are usually very weak in trees, but when we see Papilio and Diaprepes, they're not doing too bad. They're doing a little bit better. And then finally, we have the acridiids and the pterygoneads. The acridiids are the grasshoppers, and the pterygoneads are the longhorn grasshoppers. I mean, there are just at least three species of pterygoneads that are attacking citrus, and there are many acridiids that also attack it as well. And even though it's a good sign to see that because you're doing okay, the plant is still not healthy enough in order to fight them off. And so that would be in the grasshopper group right there. So I just wanted to be able to quickly give you a quick review so you can follow along and hopefully learn some of the things that I've learned over the past 25 years of being a consultant. So that will end my presentation. I wanted to get through this as quickly as possible because the authorities that be were wanted to make sure that there were time for questions. So thank you all for listening. This is another shot of Kauai, by the way. Beautiful, land. Of course, if you've been to the Hawaiian Islands, And with that, I think we will answer some questions. And I think we are now good to go. Wow, you got rid of the screen already. I was going to stop doing that, Chris, but you are on it.
You're getting lots of clapping emojis and hearts and stuff like that. People obviously really liked what you had to say. I thought it was really interesting.
Oh, so those are these little icons I see that are moving off to the right. I got it.
Yeah, like they have on Instagram or Snapchat or whatever.
Okay, I don't use those, so I wasn't familiar with that.
I don't use Snapchat, but I see like repost stuff,
Okay. Okay.
But anyway, so Charlotte Rosendahl was asking about spotted wing Drosophila in berries, and I think she's meaning brix level. Could you speak to that? And then secondly, could you there's like a whole bunch. Somebody must be just like holding the button down. Or there's a lot. It's probably both. And then secondly, could you speak to some of the solutions for bringing up brix and getting into the healthy level in terms of the level of the food that we'd want to eat to live a long life?
Okay. There are some general things that can be done. We have found almost all the pesticides, without exception, bring the brix down. We can go out there, measure the brix, and it may be low to begin with. It may be like come in at 5.8. We'll spray a pesticide and it'll go down to 5.1, 4.7. So we can see the effect of these pesticides is not helping the situation. It was a pleasure listening to Tom Murray and what he's doing, keeping things organic because it's your best chance. But even though that may be your best chance of getting a high brix, I'm going to tell you there's a lot of organic crap out there. And so because of that, there's more that needs to be done. So where do you go when you do this? I like testing. I'm a scientist, it's just kind of nice. I like to know, do you have a plant sap analysis? Do you have a brix analysis? Do you have a soil analysis? Do you have a tissue analysis? We need to find out what is going on. So in the case of Drosophila suzukii, they're going to be laying their eggs in the fruit, and it's going to need to be resilient, as Tom was even mentioning in the last presentation. So if I see that insect or other types of insect that are laying their eggs and puncturing, I'm going to be looking for, is there enough calcium? Is there enough iron? Is there enough boron? Do we have enough silica? Because these are the type of things that add hardness to a cuticle. And you're going to be short. You may be short all four, but these are the type of things that you're going to find when you have Drosophila suzuki attacking, is that there's going to be a shortage, and there's going to be mineralization, which is going to be lacking. This lack of minerals is going to lead to a soft fruit. The soft fruit is easily punctured by Drosophila suzuki. And so these are some of the general things that can help. It obviously applies to all other insects as well, whether they're eating or ovipositing, but that kind of gives you an idea of some of the things. So I like to take a look at the tests that come in and try to zero in on what the problem may be. Sometimes it's amazing. We find an absolute problem with cobalt, and that's something that nobody ever thinks of. But the cobalt comes in at rock bottom low, and we have disease in insects all over, let's say the orchard, and something needs to be done very quickly. Well, it's easy to do because you don't need that much cobalt in order to correct the situation. But these are the type of things where I like to know what we're short of and try to help the plant as fast as possible.
Speaking about cobalt, if we could just go into that, because that's something I only hear about occasionally. Do you like addressing for cobalt and other micronutrient deficiencies? Do you prefer using various rock minerals, or do you prefer using kelp? Because there's been this long debate between the two. I have some opinions about it, but I'm curious what you think.
Yes, both good. The reason why there's a debate is because both of them work. Which one works better probably depends upon your part of the country and maybe the plant that you're working on. But I have no problem with kelp, and I have no problem with micronutrient application. I have no problem with chelating agents. All of this stuff is potentially good. Sometimes it works better than other times, but yeah, you just mentioned two beautiful possibilities right now. So I don't have any problem with that. I mean, if you're spraying the pesticides right now, you've now got cobalt and manganese, a lot of things being tied up, and they're going to prevent them from getting into the plant. But you've got some good natural substances that should work. And if you're not sure if they work or not, do a brix test, apply it, and then come back the next day and see if the brix goes up. If it does, the plant likes it. If there's no jump, then there may be some other deficiency that needs to be addressed.
So speaking of micronutrients, I think just two things to elucidate people about. One is maybe if you could give a definition of chelation so people understand that, and then two, because I think that's an important concept that people when I talk about that they overlook what I say and then the second is do you know what I know, like a lot of these micronutrients are still being researched to figure out exactly what pathways of metalloproteins and whatnot that they create. But do you know if it's a metalloprotein that it creates, or is it a catalyst for reaction? What role does cobalt play?
Yes, it's both of them again. Let me address chelation first. Chelation, in the easiest sense of the word, if you have a micronutrient and it is chelated, that means it's grabbed, held onto, and is not available to the plant. And you're going to have to wait for whatever substance that grabbed it. Glyphosate's really good at grabbing onto things, especially manganese, and it will let go at some point. It may take a few months or a few years to do that, but if you've got something that is chelating something, it holds on to it really well. Chelation is a process, though, that we also use to our advantage, although it can hurt us, by separating some of these micronutrients. And so chelating agents are often used in micronutrients to keep them bioavailable to a plant. So we do that to make sure that it doesn't get tied up. So there's a little bit of back and forth when it comes to chelation. But to answer your other question, yeah, I mean, I'm still reading papers right now. I just read one from a couple of years ago that's talking about the massive number, in biochemical studies. They're still trying to figure out what some of these things are doing. Are they used as cofactors? Yes. Are they being used as cofactors in the plant? Sometimes yes, sometimes no, sometimes they're minor, sometimes they are, as you mentioned, not just being used as cofactors, but they're being used as metalloproteins. Metalloproteins will be used in order to influence good things in a plant, especially if the metalloprotein is good for the plant. So all of that is good. I mean, you're giving me a choice between two good options, Chris. So you're not giving me the choice of, would it be better to put down nutritional supplements on your plant or chainsaw it? Eat something easy that I can answer. You're throwing me softballs there. So,
well, let me feed into Tom Murray. Speaking about chelation, so Dr. Tom had brought up a major chelator. It was actually an industrial chelator, but now used in herbicide, glyphosate. And so a lot of us are dealing with, most of us are dealing at this point with soils that have been chemically abused in some way, in some time in the past. And so just to feed into Tom Murray, and then we'll come back to you, Dr. Tom. Tom, there was a question earlier. What kind of biologicals do you use? What kind of biological sprays and stuff like that? Because biologicals are something that helps break up that chelation and also helps provide good biological films, et cetera, et cetera. But Tom, could you address that? Tom Murray?
Yeah. Well, so I do brew compost tea on the farm, and I've gone back and forth from, purchasing in really high quality compost that's been lab tested. It has a full analysis with it and guarantee, to just making my own on site, with whatever debris that I have that accumulates, and either or, I've always noticed really good results. I've had people who are of the opinion that, like, hey, when you're making it on farm and you don't have all of the ingredients, exactly dialed in, and you're not getting it lab tested to know exactly what you're putting on, then maybe you're running the risk of actually introducing certain disease pathogens that are, in the compost itself or whatever. I kind of said, well, maybe that, sure, there's some logic that you could, theoretically, I could be doing that. But if that was there, I'm counting on the fact that there's so much more diversity in the compost, so that's going to negate any of those other potential pathogens that are there and really activate the soil microbiome. But that's kind of shooting in the dark, I guess. It's like versus precision agriculture, which some people are big proponents of, where I'm a little bit more like, well, how would we do it if we didn't have access to precision ag, that can be costly, even on an organic, level. It's like high quality stuff, and there's a lot of work that goes into that and those inputs. It can be costly for a small micro operation, and you need to learn. For me, I try to like hone my intuition on the farm, to tune into the farm in a greater way, and get to know your compost. Composting is one of the most rewarding activities for me over the years. I swear it's the most therapeutic thing. Turning the compost in the fall, coming back, checking it in the winter and the spring. Is it hot? Oh, it's hot. Does it smell good? Does it, all those things. It's so rewarding. And there's, just like I said, an intuitive quality to it. And I would say that I've never really used any of the on-farm compost tea that I've made, either as a foliar spray or as a soil drench that has resulted in any sort of detrimental impact to the health of the trees. It's always seemed to be beneficial and brings on a glow, and a vigor, both to the understory, to the grasses and the weeds there, and also to the tree growth. So that tells me that I must be doing a good thing by enhancing that biology, whether I have a perfect soil profile of all of the microbiology in there or not. I'm still doing a good thing, and I can see that in the response. That being said, that's kind of the on-farm. I do use a few products. I've been using a product called Pacific Grow. Minimally, it's more of as a biostimulant. Some people may be familiar with that. It's kind of a crustacean, fish crustacean kind of a slurry that's like cold processed so that all of the active enzymes and amino acids and everything are fully intact. And so, and I say I'm using that more as a biostimulant than like as a full-on fertilizer, because I'm using it at a very micro rate in conjunction with the compost tea to kind of bring a little bit more diversity there and kind of feed the microbes or act as a catalyst on their journey to where they need to get to go, to help their motility in getting into the soil profile and whatever. So that's another biological that I'm kind of using because it's so active. It's kind of considered more of a biological than just standard fish fertilizer. So yeah, those are the two main things that I'm doing. It's pretty simple, straightforward.
That's excellent, Tom. And before I come back to you, Dr. Tom, because there's a bunch of questions too, I want to address something from earlier that somebody asked, what kind of pruning tools do you use? And there's the running joke that the real orchardist has it in their pocket at all times, which neither of us do right now. But what do you like to use for pruning? And maybe if you could something that I think you and we both talked about is type of pruners and that people that use loppers or saws in the wrong way and they smash up the bark and then create a vector for disease. When to use a pruner, when to use a saw. I know that's a whole discussion in itself, but maybe just give people a couple minute primer on that and kind of what tools you actually like to use. It could be brand or type.
Well, I really like Baco. I think that they make great quality tools that really endure the test of time. Of course, I use some Felcos, but some of the pruners that I like to use most in the field, used off and on for years, have just been the standard field edition of the Bacos. They're like $20, pruning shears, versus a $50 or $60 pair of Felcos, and they work great, and they don't fatigue my hand, and the blade is strong. So I really like Baco. Really make sure to have loppers that have got a nice rubber stopper on them to catch that kinetic energy and keep it from going into your joints. I've seen some terrible loppers that have metal connectors right down there, even one with the bumper that is kind of bolted in. So you've got a little spring cushion bumper, but yet it's hitting metal on metal in that connection. If you do that for years and years, you're going to feel it in your joints. I've noticed that. I use mostly hand tools though. And so I don't really use, I don't have any power tools necessarily, although I'm really excited about some of the battery operated technology that I've seen coming out. Especially with the little chainsaws. I think that those are brilliant. I wish we had those before. It really saves a lot on the pollution factor. But yeah, I don't have any pneumatic pruners. I've thought about getting them over time, but my operation isn't really that big. And again, it would ultimately be hooked up to the tractor or more equipment. So I don't really want it to be that loud. There's a time of year or when I'm out there in the orchard, I don't really want to hear machinery in the background. I would rather be working, when I'm getting close to the trees and trying to tune into them. I don't want a motor going on in the background the entire time I'm out there. It really kills the experience for me.
Oh yeah.
I can imagine having that on a day-to-day basis. Yeah.
Yeah, that perennial weed whacking all day long. It's really terrible.
Yeah. Yeah.
Dr. Tom, and I think you could talk about this too, Tom M. But can you talk about? There's been a lot of questions about one. Dr. Tom, could you talk about the plant presses that you use to extract juice and what you like to use? And then number two, can we talk for a minute about mineral analyses So I'll start with Dr. Tom. But where do you like to go get your soil tested? If you're just starting out, you're wanting to address brix from a nutritional standpoint. Go to the roots of the problem, as it were. What do you recommend?
There were several questions that you just asked.
Yeah.
I'm not sure which one to deal with first. So,
How about plant press?
Plant press.
hydro?
Spectrum Technologies was my favorite, which they discontinued. So it's easy for me to recommend it because I'm not obviously going to be making any kickback on stuff like that. So they discontinued. That was my favorite. It was the one that was off to the left that we saw up there. It's got a bigger container. You can put more leaves in it. It's not as powerful. It doesn't completely pulverize the leaf, whereas the one on the right that I had listed up there is a little too powerful for me, and I don't need to pulverize the leaf. I'm just trying to get plant sap out of it. But I can work with it. So any of the plant saps that are out there, I can presses that I can work with. I prefer not to use my hands, especially because if I'm doing dozens of readings, my hands are going to get tired, and I might be pretty strong on the first, second, and third, and fourth one. But by the time I get to 25 and 30, I'm going to get tired, and I'm not going to be able to squeeze out as well. So using a plant press is really the only way that I'm going to work. So if Spectrum is listening right now, you're welcome to start up production again. I would appreciate it. As far as the micronutrients, where can you get it? I think it's best to look at the plant directly, and that's why I want to see a plant sap analysis if we can. Obviously, I'm with AEA, so I'm familiar with those. I can work with the tissue analysis. The soil analysis is helpful too. Just want to know what's available, what's in the soil. And you can usually pinpoint, especially if you're attacking it from the top and the bottom of the plant, you can usually pinpoint as to what needs to be taken care of, because it's almost always a micronutrient problem that is causing the issue. So I think I just answered 2 questions, but you probably asked 9 of me. So I think I've got 7 more. Yeah.
I think you integrated it well. One little follow up here, and then I want to ask Tom Murray a question. But when do you sample the leaf? When do you sample the soil? I think some people might be wanting to know that.
Yeah, the leaves I prefer to do when they're at maximum photosynthesis. So I prefer to go out in the heat of the day, which I'm sure is just disgusting. So many people thinking, I cannot believe you're going out in 96-degree weather in order to do it. But when the sun is out and it's hot, yes, I go out there with a canopy. I put out a table. I put my plant set press on it, and I'm going to go at it probably even for a couple hours. So yeah, I'm going to be somewhat protected. It also protects my refractometer, because if I'm using that thing in full sun, it will turn off on me, which is just a safety mechanism for it. But yeah, that's what I do. I do it, and I test them within 5 minutes after I get it. Some people do it 3 hours later, and you're going to get very different readings. They're going to be much higher than they're supposed to be. But you need to test it. I test it right there on the spot. So I'm set and ready to go, and that's what I'm going to do. So I prefer to go out there at the maximum photosynthesis. I just want to know what's the plant doing at its best. I don't want to know what it's doing at its worst. I want to know what it's doing at its best. And if it's in full sun right now, that's going to be its best. It's going to be photosynthesizing well. And if that's at maximum photosynthesis and I'm getting 5.4, I'm thinking, wow, this is the maximum. This is as good as it gets, is it? And that tells me a lot that this plant is in trouble and is going to need it. And so doing the brix refractometer is very easy, because many of the farmers and the homeowners can use it. You put power into your hands. You can find out a lot without spending a lot of money and sending it out. However, when you do spend some money and put it out, that information is also useful as well, because it helps to pinpoint as to what the problem is. Otherwise, you're just finding out what's healthy, what's not healthy. And even though I've got a pretty good idea of where things are on the brix scale, I do prefer to have a little bit more information when I'm doing it. So yeah, I go out in the afternoon. That's when I prefer to do it, but not when it's raining.
Yeah, a little follow up. What kind of where would you take leaves from? What would be a good sample of leaves? There's a question about that.
Yeah, I just take them from the middle, because I think we all know that there are changes from the flush, and there are also going to be changes on the leaves at the bottom. So when I'm doing a brix test, I'm just going to pull from the middle of the plant, and I need 30 leaves. In order to satisfy statistical vigor, I'm going to need 30 leaves to do it, and those 30 leaves I'm going to pull from 30 different plants. So if I'm sampling the field, I'm going to pull from 30 different plants, one leaf from each, and that's going to give me an idea of what's going on in that field. With a citrus tree, if you want to zero in, you can easily pull off 30 leaves from a citrus tree, and you can zero in on a particular tree if you want to. But when I'm out there, I'm just looking for the general consensus, and I want to know how things are doing in a field. And so I want to be able to get that, and to go out there and collect as many leaves as possible. I don't want to overdo the plant. I want to take 30 from the same plant if I can help it. Some trees can handle it just fine. But if there are a lot of young citrus trees, we're not going to go get 30 leaves because they may only have 200 leaves on them, and you're not going to take 30 leaves off if you can help it, especially if it's growing fast and it needs that nutrition. So usually one at a time. And if I'm going out there frequently, like once every week or every 2 weeks, this puts less stress upon the fruit trees because they're not losing too many leaves.
Mm-hmm.
Chris, we lost you for a second, but I know you're going to come back. I just want to make sure that both our speakers can hang with us. We've got some great questions, so we can go over until about 6:40 Eastern time. So probably another 5 minutes or 6 minutes of Q&A. Is that all right?
Yeah.
With you?
Sure.
That'll be fine.
I hit the mute.
Great.
I
Okay,
hit the
good.
mute. Sorry, Tom.
Oh,
Tom,
no worries.
could you address what you do for soil testing for folks, and also deer protection? Could you talk a little bit about deer protection? There's a question about that.
Oh yeah. So, well, soil testing, I've just been basically doing a general mineral analysis. I don't really, I'm not big on adding nitrogen inputs. With all of my experience, the trees will do just fine without loading them up with nitrogen, and they'll stay pretty darn healthy. And I've noticed negative effects in the orchard and or other people's orchards when they use nitrogen, especially in excess. So I don't ever test for that. I just do a general mineral analysis, and I've been using. Drop a name here of a lab that I think is really great, Perry Agricultural Lab in Montana. That would be PAL as the acronym. They're great. Been doing it for 30 some odd years, and it's just really easy to read, and the recommendations are really good. So I'm really dialing in. One of the things that I focus on when I'm doing the soil analysis and the minerals, specifically looking at the calcium, the cal-mag ratio, as the traditional Albrecht kind of model is kind of what I've focused on, and then in conjunction relying on the microbes to kind of assist me in that, and then also boron has been critical. I've had issues with boron, and we do in our soil because we get so much rain here that I have used that as a supplemental. And I found that really, really helpful. So those are kind of like the three that I really am concerned about when I'm testing the soil. And yeah, I'm pretty balanced right now. The last soil results that I got back, and I took, I did quite a few more in some other areas of the orchard that I haven't looked at in a while. And I was really pleased with the way things are holding in that sense. And again, I do, part of the biological compost tea thing is because I really adhere to kind of like Elaine Ingham's work with the Soil Regen School, and one of my mentors actually studied with her when she was at OSU, and I really like this kind of goes in line with what Dr. Tom had mentioned, in his slide presentation, of Kauai, and showing us like, here is a beautiful lush habitat. There's plenty of insects out here. I mean, there's countless amounts of insects, but there's plenty of healthy, vigorous plant life out here that is not succumbing to infestations. We're not too far away, so there's a balancing effect there. And what is that? It seems to have to do with the foundational piece being the soil biology. And that's kind of resonates with Elaine's school of thought, which is kind of where I've come from, is that if you take care of the soil and the biology, and think about not feeding the tree, but feeding the biology of the soil, and take care of that first and foremost, then you're going to have a pretty healthy, resilient system. So that's kind of like how I approach it. And then I do check in with the minerals to make sure that there's enough there in available form through the soil testing that I do. To the second question, deer, I've noticed that any of those kind of the animals that come through the orchard seem to come in different cycles. We are next to a huge wildlife refuge. They're not far away. We're right on the road though, and it's a pretty busy road, and I think that there's plenty for them. They kind of stay away from the road. We also have a resident, I've had bigger problems with ground squirrels. California ground squirrels have moved in. They're an invasive pest that is really tenacious. And luckily, I actually have, fortunately, I have a vermintor that lives on the farm who's a young kid who's a really good shot. And yes, I do pay him by the head. And I've heard from elder farmers when those pests moved in. I'd heard about them for years, and I said, "Hey, these little ground squirrels showed up," and they said, and some of these people are really devout spiritually and would not harm anything, wouldn't kill anything. But as farmers, they were like, "You have to do something about those. They will dig under your house. They will eat your wires of the house. They will." I mean, these things
destroy everything.
They will proliferate, and I've seen that. The first year we saw one or two. About 18 months into it, he had already taken out 80 of them, and we still have him. He's knocked the population down quite a bit, but I've been to some other friends' farms that didn't do anything about it, and they're having a really hard time establishing any crops because they're so prolific. It's really, now deer seem to kind of stay away because I think that there's various factors, and they know that he's running around there, and yeah, he has a deer tag too. When we had problems in the past, we said, hey, if they keep eating the young trees, I'm not really excited about going and taking out the deer. I kind of like to leave nature alone and create a space for them on the farm. So, but when they start eating all of the baby trees that we just planted and stuff, it's like, well, maybe we'll go get a tag and let them know that we're having pest issues. And so that's kind of how I deal with it. I don't have a magic deterrent.
Sorry, Tom.
Other than that,
we got to wrap it up. We went over a little bit, but I think it was worth it. I think everybody appreciated it. There's still actually a bunch of questions for the Toms, both the Toms. So go Team Tom, and I appreciate everybody showing up today. And I did want to mention that that this recording will be made available on nafex.org in 1 to 2 days, and then in about a year on NAFEX TV. And that you should check out, because there are on most presentations other supplemental content that you can review, and you can review that at any time. It will stay up there. And then don't forget to check out our other sessions that are coming up. And also, again, thank you for joining us. So take care, Tom Murray and Dr. Tom. So thank you for your time.
All right. Thank you.
Yeah. Thanks, Dr. Tom. That was great.